Good morning, students. I welcome you all onto this online class on nuclear polyhydrosis virus, which is commonly called as NPVs. In my previous classes, we had studied about the other different types of bacterial as well as fungal biopesticides. In this particular class, let us study about nuclear polyhydrosis virus as a biocontrolling agent towards various pests. So, as we all know, there are various different kinds of biopesticides has been produced all over the world, out of which bacterial products has been taken a global share, highest global share in the market. Bacterial products, especially bacterial thuringiensis, has taken up the highest production rate in the global market. This is followed by fungal products, especially trichoderma has played a major role, where 27% of the fungal products has been used extensively all over the world. Next comes the viral products. The viral products, there are various advantages as well as disadvantages with respect to the viral products. One thing is the production is cost effective. The other important thing is this viral products gets mutated to that of a UV rays, that is the major drawback of this viral products. One thing is they are cost effective. The production requires large amount of the infrastructure as well as the money for the production, and also because of the mutation rate is very high. That is the reason the preference has been given to that of bacterial products, followed by the fungal products, and next comes the viral products, and also the other nematodes also have been used extensively as a biopesticides. So, however, today let us study about nuclear polyhydrosis virus. What are these nuclear polyhydrosis virus? How they have been classified? On what basis they have been classified? How they have been named? What is the nomenclature involved? How actually they have been named? On what basis they have been named? Let us study in detail. And the mode of action of these NPVs. Today we are going to concentrate only on NPVs. We are going to study the mode of action, how actually it destroys the pest, and also the life cycle of this NPVs. We are going to study the production of these NPVs and the advantages and disadvantages of this nuclear polyhydrosis virus in detail we are going to study, and how it has to be applied onto the field. These are the different aspects we are going to study in this particular video lecture. So, before going into the detail of these NPVs, what are these virus? So, virus we all know, not even the life science students nowadays, because of this COVID-19, most of the people, even the common man, know now what these viruses are. COVID-19 has taught the lesson to everyone. So, we know the viruses are the group of organism which shares the characteristic feature of both living as well as non-living things. These, when they are present inside the host, they are always parasitic, where they reproduce only when they are present inside the host. Always they are obligatory parasite, and they reproduce only when they are found inside the host, and when they are outside the host they are just like a non-living thing. And we all know it is mainly composed of a nucleic acid and the protein. These are the two important component in these viruses. So, however, viruses are submicroscopic, intracellular and obligate pathogenic entities with nucleic acid and proteins. They are the genus and species specific. Actually, they are genus and species specific. However, we know this COVID-19, which we know that this is one of the virus which is found to be specific to that of a bats, but maybe due to some mutation, it has also infected a human. I don't know how this sentence is apt in this particular situation. They are genus and species specific. Some of the viruses are found to be potential of controlling pest. They are the group of viruses such as baculovirus, cytoplasmic polyhydrosis virus, entomopox virus, iridovirus, densovirus, small RNA viruses. These are some of the group of viruses which are found to be extensively used in controlling the pest. So, now let us study in detail out of all these viruses, baculovirus is a group of virus which has a broad spectrum of activity and it has been extensively used in controlling the pest. So, here we are going to study baculoviruses in detail. So, what are these baculovirus? How they are? They are a group of virus which have been exploited widely as a biocontrol agent. These baculovirus are rod-shaped enveloped virus with circular double-stranded DNA genome of 80 to 180 kilobase. This you have to remember, baculovirus, they are rod-shaped enveloped virus with circular double-stranded DNA. Remember, this is one of the DNA virus, double-stranded DNA virus, and it is about, the genome is about 80 to 180 kilobase, that you have to remember. And they belongs to the family, this baculovirus belongs to the family Baculoviridae. All the organism, the virus belongs to this Baculoviridae. They are rod-shaped viruses and they have a circular double-stranded DNA inside them. The family Baculoviridae has been divided into two subfamily, Eubaculoviridae and Nudibaculoviridae. So, here in Eubaculoviridae, it consists of two genera. You can find two genera in this Eubaculoviridae, that is nuclear polyhydrosis virus, which is commonly called as NPV, where the occlusion body, I am going to tell about what is this occlusion body and all later. Occlusion body consists of the crystalline matrix composed of a protein called as polyhydrin, that you have to remember. So, here the nuclear capsid has been enclosed over by a proteinaceous matrix. That proteinaceous matrix is made up of polyhydrin, that is occlusion body. Inside these occlusion bodies, you can find nucleocapsids. Rod-shaped nucleocapsid can be found. These nucleocapsid has been enclosed by a protein called as polyhydrin, that you have to remember. That is the reason it has been called as nuclear polyhydrosis, because the presence of a protein called as polyhydrin, which forms the matrix for the inclusion bodies. That is the reason it is called that it have been placed under nuclear polyhydrosis virus. There is one more genera under this Eubaculoviridae, that is granulosis virus, where here the protein, that is the protein which covers this nuclear capsid is made up of granulin protein. That is the reason it has been placed under granulosis viruses. Polyhydrosis virus, granulosis virus. Polyhydrosis virus, because of the protein which they have, it is called as polyhydrin. Granulin protein can be observed in granulosis virus. There is another group of viruses which belongs to the subfamily Nudibaculoviridae. It consists of only non-occulated viruses, NOB, without occlusion body formation. In case of Baculoviridae, you can find occluded bodies, that is a nucleocapsid virus, that is rod shaped virus has been enclosed by a matrix, proteinaceous matrix, where in case of polyhydrosis or granulosis virus. Whereas in this Baculoviridae, no such occlusion body formation happen, no such matrix is been found. That is the reason they have been placed under Nudibaculoviridae. Sorry. So now let us move on to NPV. Let us study the structure of these NPVs in detail. NPVs, how these NPVs have been named. So, before going into these NPVs, let us study the structure of these NPVs. So, here in this particular image, you could find some occlusion bodies, that is OBs, they are called as OBs. What are these OBs? These are crystalline structures. So, you can see the structure which is in the form of crystal, and inside this crystalline structure you can find nucleocapsid rod shaped viruses you can find. They are called as occlusion derived virus. You can find these are the viruses where you can find a genetic material inside them and a proteinaceous covering, that is its nucleocapsid. These are called as occlusion derived virus. These occlusion derived virus can be classified as SNPV or MNPV. What is this SNPV? If a nucleocapsid, say for example, if this is a nucleocapsid, it is single in a polyhydrin. See here, this is SNPV, where this polyhydrin consists of SNPVs, where this SNPV they are single, it is single nucleocapsid can be found in a polyhydrin envelope. In a polyhydrin envelope, you can find a nucleocapsid which is single. Whereas in case of MNPV, the occlusion body is nothing but a polyhydrin, where it is a matrix consists of polyhydrin envelope. Inside this polyhydrin envelope, you can find the nucleocapsids. These nucleocapsid can also be called as ODVs, occlusion derived virus. In case of MNPV, you can find more in each thing, you can find the nucleocapsid or more than one, say there in each of the matrices you can find more than one nucleocapsids. More than one nucleocapsid are present in occlusion derived viruses. These are occlusion derived virus where you can find many ODVs in OBs. See, each one of these crystalline structures are called as what? OB. What is OB? Occlusion bodies. Occlusion bodies may contain many ODVs. ODVs are nothing but the nuclear capsid. This nuclear capsid or ODVs can be single as in case of SNPV, or it can be MNPV, that is there can be more than one nucleocapsid can be present, then it is called as MNPV. Remember, SNPV where the polyhydrin inclusion consists of a nucleocapsid made up of which is single, whereas in case of MNPV where you can find the ODs, occlusion bodies consist of many ODVs, where in each ODVs you can find many nucleocapsids. Those are called as MNPVs, remember. And you can also find here in this particular image budded virus. They are called as BVs. What are these budded virus? After the process of the replication after the process of assembly, of a genetic material as well as a proteinaceous covering or capsomeres, later when they have to emerge out, they emerge out as a budded virus. This is due to the where they have to release from that of a host cell. During that particular process, the cytoplasmic membrane of the host may help in the synthesizing of this particular protein around, proteinaceous covering around this particular nucleocapsid. That is the reason it is called as budded virus. During the process of the release of this virus, they come out in the form of budded virus. So, they are called as BVs. So, this is the structure of NPVs. Once again I will repeat, NPVs are nothing but nuclear polyhydrosis virus. They are called as nuclear polyhydrosis virus because their replication happen inside the nucleus of the host cell, and they are called as polyhydrosis because the occlusion body, the proteinaceous matrix is found to be made up of polyhydrin, and these viruses usually they are made up of a nucleocapsid, and it is rod shaped virus, and it is made up of a DNA which is a double stranded circular DNA, and we can find SNPVs, MNPVs. SNPVs are the one where single nucleocapsid in a polyhydrin envelope. MNPVs, if the nuclear capsid or more than one in a polyhydrin envelope, they are called as MNPVs. And how this nomenclature of this NPVs has been done? According to International Committee of Taxonomy of Viruses, ICTV, 4 genera has been recognized. They are alpha baculoviruses, that is Lepidopteran specific NPVs, alpha baculovirus. They are specific only to that of a Lepidopteran larva. So, they are alpha specific NPVs. Beta baculoviruses, Lepidopteran specific GVs. GVs, why it has been named as GVs? Because they have been made up of a protein called as granulosin. Gamma baculovirus, they are specific to Hymenopteran specific NPVs. They are specific to Hymenopteran, that is in insecta you have many classes where they are specific only to Hymenopteran species. You have delta baculovirus, they are diphtheran specific NPVs. They are specific only to the insecta, that is the class Diphthera. So, you can classify, or you can find 4 genera has been recognized according to ICTV. What is this ICTV? Remember, for the point of your competitive examination, International Committee of Taxonomy of Viruses, ICTV, 4 genera has been recognized: alpha baculovirus, beta baculovirus, gamma baculovirus, delta baculovirus. So, next let us study about the naming of this, how this naming of this NPVs has been done in my next slide. Nomenclature of baculovirus. Traditional named after the insect species from which they were isolated first. So, usually the nomenclature is nothing but naming according to ICTV. It can be named traditionally how, so where, from which particular insect they have been isolated from. That is important. For example, Autographa californica NPV. This NPV, why it is MNPVs? Because you can find more than one nucleocapsid in an envelope, in a proteinaceous envelope. So, it is called as MNPV, remember, and it is Autographa californica. This particular virus is called as Autographa californica MNPV because it has been isolated from a insect called as, or which belongs to the genera Autographa californica is the species, and they are MNPV. So, it can be abbreviated as AC, capital A, small c, MNPV. Likewise, you have Bombax mori, MNPV. Bombax genus, M is mori, species, NPV. Likewise, you have Helicoverpazia, SNPV. This is one of an NPV which has single nucleocapsid has been enveloped in a polyhydrin matrix. So, they are SNPV and it can be abbreviated as HZSNPV. H is because the genus of the insect from where it was isolated was Helicoverpazia, species is Zia. SNPV because the NPV is having a single nucleocapsid in a polyhydrin matrix. Likewise, we have Spodoptera lutura MNPV which has been abbreviated as SLMNPV. So, likewise this has been named, but remember for the point of examination in your competitive examination they can ask about the nomenclature where it can be named as for the species from where this particular virus has been isolated for the first time and what kind of the NPV they are, whether they are multiple or single inclusion bodies they are, based on that it can be named. 15 NPVs have been identified with 483 species. There are 15 NPVs has been identified with 483 species till date. So, this is about the nomenclature of baculoviruses. So, now let us study the mode of action of these NPVs. How actually these NPVs destroy or infect the pest, how actually they destroy them. So, in this particular image you can find a Lepidopteran larvae, and here we have elaborated the gut region, the epithelial cells of the gut of this particular insect. And in this particular image, I have shown how actually the replication of this virus happen. So, let us study this in detail. For example, if a Lepidopteran larvae feed on this particular NPV. Say for example, if we have used this NPV and if it has been sprayed onto the leaf, when this larvae feed on these leaves, along with the leaves, it can also engulf the occlusion bodies, that is the crystalline structure, polyhydrin crystalline structures which have been present on the surface of the leaf. It is also being engulfed by this particular Lepidopteran larvae. Once it enters into the midgut region, if they enter into the midgut region, there the inside this particular midgut region, as we all know, the pH is alkaline. Because of that alkaline condition in this gut region, what happened? This polyhydrin or the occlusion bodies which has been enclosed over by that of the envelope, that is the protein called as polyhydrin has been enclosed. Because of this alkaline pH and also because of the activity of an enzyme called as protease, because of the activity of an enzyme protease, polyhydrin has been digested by this particular enzyme and also due to the alkaline condition in this particular region of the gut. So, what happened? So, what happened? So, whatever the obese, that is occlusion body, which is having many nucleocapsid inside them, they are released. They are released. See, for example, here you can find occlusion body. So, this is the occlusion body which is crystalline because it has got many obese ODVs inside them and it has been enclosed over by a polyhydrin matrix. Polyhydrin matrix. Because of the pH, alkaline pH, and also because of the enzyme activity, especially protease enzyme has been produced in that particular region. This particular polyhydrin is being degraded or it has been digested by these enzymes, wherein which ODVs, occlusion derived virus, you know, occlusion bodies has got many ODVs. These ODVs are released, they are released inside this midgut lumen. So, this is the midgut lumen. See, this is the leaf, you have occlusion bodies. Occlusion bodies, if an insect feed on that particular occlusion body, it enters into this midgut region. In this midgut region, what is happening? That is what I am explaining. So, this occlusion body enter into the midgut lumen, where due to the enzymatic activity and also because of the pH, the polyhydrin is being decomposed or they have been digested, which releases ODVs, occlusion derived virus. All these were there inside. These ODVs are released into this midgut lumen. Later, what happens in this midgut lumen? This viruses as well as the host encoded enzyme. The host encoded enzyme present in these ODVs, they are the one which decompose or which digest this peritropic membrane. So, you have a peritropic membrane, this is the gut lumen and this is peritropic membrane. So, this if the ODVs has to enter into the host cell, even this membrane has to be decomposed. So, the way has to be there to enter. So, that is the reason ODVs encoded enzymes by the host will this enzyme are the one which decays or digest the peritropic membrane. This peritropic membrane is being degraded by the enzyme which have been encoded by the host as well as the virus. So, once they have been degraded, later what happens to this particular ODV? Sorry. What happens to this particular ODV? So, this particular cell now I have elaborated over here. See here the peritropic membrane was there. So, this has been digested. So, now there is a way for this particular ODV to enter inside. So, how they will enter? This ODVs enter into the cell by the fusion with the epithelial cells microvilli. So, this is one epithelial cell and these are the microvilli. These are microvilli. So, what this ODV does? This ODV fuses, made up of, this is also a protein, isn't it? So, this fuses with that of a epithelial cell, especially with that of a microvilli. It fuses with the epithelial cells of the microvilli and the nucleocapsids are released. These are released into the cytoplasm. They are entering into the cytoplasm of the host cell. Later, after they enter into the cytoplasm, they even enter into the nucleus. They enter into the nucleus, that is why it is called as nuclear polyhedrosis virus. They enters into the nucleus. Later what happen? They undergoes the process of disassembly, that is the proteinaceous component as well as the nucleic acid components are disassembled. They are disassembled, and later what happens? The DNA of this virus, they transcribe and they translate the necessary proteins. The DNA of the virus now, they transcribe and translate the necessary genes, early genes and late genes, which are essential for the process of the replication of the DNA as well as the synthesis of the proteinaceous sheath or the proteinaceous covering. All these genes are transcribed and translated. You can see over here, translation, transcription, mRNA formation, all this happen later. All these and all this whichever the proteins have been synthesized, they all they may be synthesized in the cytoplasmic region, as wherever it is, they are synthesized in the cytoplasmic region, and they are the one which are mainly involved in the process of the formation of this virus. Once all these enzymes or the proteins which are necessary for the process of the replication of the virus, even they have been produced inside the cytoplasmic region, and later they enter into the nucleus of the host cell. There again they assemble together, that is assembly of the DNA as well as the protein happen. Once they assembled, they are released into the cytoplasmic region, and once they are released into the cytoplasmic region, they enter into the to the cytoplasmic region, and they go towards that of a basal membrane of an epithelial cells, and there by the process of budding of the epithelial cells by the cytoplasmic membrane of this particular cell, where they are involved in the process, the formation of budding viron, where actually the viral envelope fusion protein has been synthesized, and due to this particular process the virus that is BVs are synthesized. These kind of BVs are synthesized, that is budding virus. This is called as budding virus because of the how actually they come out, because by the process of the budding of this epithelial cells. During the process of this budding, what happens, this viral envelope fusion proteins are synthesized, and they are the one which are involved in the formation of such polyhedron matrix, and since just they are formed in such a way, so they are called as budded virons, they are called as BVs. So, at that particular time what happens to that of the host cell? Because of all the activities which is happening, all the transcription and translation of the proteins which are required for that of the viral virus synthesis is happening. At the same time, this cells, the host cell may not undergo the process of any metabolism, and they and also the lysis of this particular epithelial cells is happening, and also there is no process of absorption of the nutrition is happening. That is the reason at that particular moment the insect cannot digest the necessary food materials, and also they cannot absorb the nutrition which has been digested, due to which the insect insect starve and it will die. Let us study how actually the symptoms and all. Once again I will repeat the mode of action. Once the insect larvae or the insect pest feed on this kind of OBs are nothing but occlusion bodies. Once they feed on this OBs, once this OB enter into the midgut region, due to the alkaline pH and also due to the enzyme protease, the polyhydrin is being digested, so that ODVs, occlusion derived viroids are released. These occlusion derived virus, they digest, and also due to the enzymatic due to the host encoded enzyme, virus and host encoded enzymes are coded, and these enzymes are the one which decomposes the peritonic membrane of the midgut, and finally these ODVs, they have to enter inside the host cell. How they enter? They can enter by the process of the fusion of this ODV with that of a villi, microvilli of these epithelial cells. Once they fuse, the nucleocapsids are released inside the host cell, and these nucleocapsids, once they enter into the cytoplasm, they will enter into the nucleus. Once they enter into the nucleus, they translate, and first they undergo a process of they get disassembled, that is the DNA as well as the protein gets disassembled, and at the same time the DNA of the virus encodes, translates and transcribe early and late genes which are responsible for the synthesis of this virus. Once they have been synthesized, the translation has happened, the proteins required for the replication of the virus has been synthesized in the cytoplasm region. We all know all those transcription, translation and all. After the protein has been synthesized, all the proteins which are required for the process of replication enters into the nucleus. There the DNA as well as the protein gets assembled. After the process of assembly, the progeny moves out from the nucleus to that of a cytoplasm, and later they enter into the basal region of this particular host cell. There they undergoes a process of budding at the same time viral envelope fusion protein is also being produced, and this particular protein or the one which are involved in the formation of such proteinaceous matrix, and they have been budded out of these epithelial cells and they are released outside, and at the same time due to the less metabolic activity of this host cell, the digestion as well as the absorption of nutrient may not happen, which leads to the death of the cells. And at the same time, once this BVs, that is budded virons, are released outside, these budded virons can attack another epithelial cell, and again many progenies are produced. In the same way, all these progenies which have been produced, they can infect many epithelial cells and they can decompose or degrade the entire epithelial layer of this midgut, due to which the Lepidopteran larvae starve and it will die. So, this is the mode of action of NPVs. So, here you have given some description of the different events, you can go through that, and also the same the life cycle of this NPVs, how actually it happened. So, as I was mentioning earlier also, the nuclear polyhydrosis virus can be of, this image explains that nuclear polyhydrosis virus, it can be either a multiple NPV or a single NPV, where in multiple NPVs you can find more than one nucleocapsid, whereas in case of SNPVs, the nucleocapsid is only one single in OB, occlusion bodies. So, such OBs, occlusion bodies, which if they have been fed by that of if such occlusion bodies are fed by the larvae or an insect pest, once they feed, these occlusion bodies enter into the midgut region. So, in this midgut region, what happen? Say for example, if this is a epithelial cell, So, here in this midgut region, due to the alkaline pH as well as due to the proteolytic activity, the proteinaceous polyhydrin gets decomposed, which releases many ODVs. All these ODVs which were present inside were released. These ODVs fuses with that of a microvilli, epithelial cells of the microvilli, they fuses, and the nuclear capsid enters inside the nucleus. They enters inside the nucleus. Once they enters inside the nucleus, the disassembly happen, and later the DNA of this particular virus will transcribe and translate for the proteins, early and late proteins. Once all these have been synthesized inside in the cytoplasmic region, they again re-enter, they enter inside the nucleus. Once all these proteins are present, the DNA replication happen, and at the same time the proteins which are necessary for the process of assembly or for the process of the formation of the structural proteins, all these are synthesized and they enter inside the nucleus. Again the assembly happen, they get assembled, and once they assemble, you can find them inside the nucleus. Inside the nucleus you can find, so they are called as nuclear polyhydrosis virus, and at the same time the cell can also lyse, and once they lyse, these viral particles which has been produced, they can infect the other epithelial cell, that is secondary infection can also happen, and once the epithelial cells have been lysed and once they occupy, the nucleus of this epithelial cells occupies all these NPVs, the metabolism or the function of this epithelial cell has been lost, because of which the digestion is been interfered, the digestion of the necessary proteins, digestion of the metabolism is been affected, due to which the insect starve, and the same time the cells of the midgut also gets decomposed, and finally this virus can also spread to the entire part of the body of this particular larvae, and finally this larvae become sluggish, that is they become inactive and they die. How actually they die? That is more interesting. They go and occupy the tip of the leaf or whichever the part it is feeding on, and they hang upside down. That is an important characteristic feature. You can see this particular Lepidopteran larvae which is just hanging upside down. So, once they just starve and once they are about to die, they go and attach onto the tip of the leaf and they hang upside down. So, that is the characteristic feature of this particular NPVs, that you have to, if you find any larvae which is just hanging on the leaf or infected part upside down, indicates this is due to the activity of NPVs. So, here I have elaborated the life cycle in detail, and if you can also go and observe this particular animation which is in the form of cartoon. Very interesting, you can see that also the mode of action, you can, the link I have provided, you can go into that and study in detail, and also I have given the links for each of the images which I have put, you can go to that link and study more on that also. So, symptom of the infected larvae. So, once you can see this particular images, once the larvae, this is the healthy one and this is the dead insect. So, once the larvae feed on such kind of viral particles, they become sluggish. Sluggish means they become just inactive, they stop feeding, and discoloration of the skin. Usually this larvae, caterpillars, whatever the color, they undergo the process of discoloration, you can find the discoloration, and regurgitation happen. Whatever the food it has eaten, that particular insect has eaten, it will again come out to the mouth like one vomit. That is called as regurgitation. Regurgitation happen, the food which it has fed, due to the action, those food material will never enter inside the gut region. To the infected larvae I am talking about, it again come back to the mouth, that is called as regurgitation, and it produces wet dropping, and also this insect die. they just decay like this. Or you can find the insects, especially you can see these insects where you, in case of chickpeas in India, the characteristic feature, characteristic hanging you can find where they die and just hang upside down like this, upside down. That is the characteristic feature of the insect which has been infected by NPVs. And the symptoms, the larvae, once it feed and once the mode of action happen inside the gut, these insects larvae become sluggish. They undergo process of discoloration, see check the spelling over there, discoloration of the skin, regurgitation, wet dropping, and finally the insect decay, and also they can die by just hanging characteristic hanging where actually you can find them upside down. So, these are some of the symptoms of this Lepidopteran larvae. So, how this production of NPVs can be done? So, you know the virus, as like your bacteria or your fungi or any other microorganism, it cannot be produced as such, it cannot be grown in vitro like on artificial media and all. So, in vitro condition we require some infected cells, since they are parasites we require the host, the infected cells cultivated in bioreactor. That is, many insect cell lines have been developed for in vitro study for mode of action and multiplication of NPVs. That is, cell lines of this Lepidopteran larvae can be brought, and these cell lines are allowed to infect with that of NPVs, and multiplication of these cells can be done. That is, we are allowing this particular NPVs to grow inside the cells and multiplying them, and such suspension we are going to use as a biopesticide. And also in vivo condition, that is in field, how we can do infecting larvae and insect rearing technology. That is, in case of in vitro we are bringing the cell lines, and in aseptic condition we are growing them in a regulated manner. Whereas in vivo condition, like how we are going to rear these silkworms and all, in the same ways those Lepidopteran larvae or any larvae which you have to which kind of virus you have to multiply, such larvae can be brought. We can feed these larvae with NPVs. With NPVs we can feed them, and once the NPVs has been fed inside this Lepidoptera, inside this larvae, inside these larvae the multiplication of virus will happen. So, such worms or such insects are collected and they can be macerated thoroughly and diluted in water, and it can be spread, or you can just use that as a biopesticide. So, in vitro condition you are using cell lines, in vivo condition directly you are going to infect the larvae with NPVs and allowing this NPVs to multiply inside them. And such insect larvae we can collect and we can macerate that particular insect larvae thoroughly and dilute it with some amount of water. That water suspension, now it has got many viral particles. Such suspension can be spread onto the field. So, these are the some of the method of production of this NPVs. So, this is one of the table which represents the number of the baculovirus preparation which has been registered for pest control in various part of the world. So, here these are some of the products which I have listed out in the different countries and against various species of insects. In India especially Biocontrol Research Laboratory and Multiplex at Bangalore are involved in the mass production of HA-NPV and SI-NPVs in vivo condition. So, in vivo condition these are the two different laboratories in Bangalore, they are involved in the production of these pesticides. So, what are the advantages and disadvantages of these NPVs? The main advantage is NPVs are specific to target pest, they are specific to a particular pest, safe to environment and human health, eco-friendly, less residual toxicity. The disadvantages: slow in action, rapid inactivation by UV radiation and natural sunlight, that is the most disadvantage. Production cost is effective, wash away due to rain or wind, etc. So, here we cannot use any we cannot produce any transgenic plants or any other like that of BT, or using this genes, or using chitinase genes of trichoderma and incorporating in the plants and all cannot be done. It has to be spreaded onto the plantation. So, due to the rain or or wind or whatever it is, they get washed away, that is another disadvantage. And since they are virus, they are more susceptible for the process of mutation, and for the production of this also it is cost effective, for example especially for in vitro condition, and also the action is quite slow. So, these are some of the advantages and disadvantages of NPVs. So, to conclude, NPVs are being one of the most important biopesticide, as it is eco-friendly, as they have less residual toxicity, compatible with many chemical pesticide. Hence, NPV can be implemented as one of the major component in integrated pest management program. So, this is the conclusion. So, this is about the NPVs. However, this NPVs, nuclear polyhydrosis virus, is one of the virus which has been extensively used as a biopesticides, with especially to that of many insects. It has got a broad spectrum of activity and less toxicity, and this property can be exploited for the pest management program. So, this is about NPVs, and these are some of the references. Apart from many textbooks also this about this NPVs are available. I have listed out. Apart from that, these are some of the links which I have provided. You refer those links, as well as you can refer these two papers, very easily explained. These are the review papers there. You can refer these review papers also and use it accordingly. Thank you. Thank you once again for your patience listening. Meet you in my next video lecture. Thank you. Thank you all once again.